So I think most of you in this room would read Live and Learn from Shigella Invasion of Epithelial Cells. But because the topic of this conference here is live imaging, I thought that Live and Learn would actually be more appropriate. So this is just to mention that seeing is believing, of course, but you want to make sure that what you believe is close to the truth, because you might very well be taken to mistake by some awkward sentence or awkward formulation. So I think it's important that to see, to observe first, but it's also important to diversify the angles of observation so that you limit the chance to asserting an artifact, or increase the chance to be closer to the truth at least. So going back to what we're interested in, for several years that we've been interested in invasion of epithelial cells by bacterial pathogens. So here I'm just reminding you of the various strategies that are being described. One used by Listeria or Yersinia, that is mediated by the binding of invasins with strong affinity to surface receptors such as integrins or cadherins. And this is the high affinity binding that would lead to the zipping of pseudopodal membranes around the bacterial surface and engulfment into a tight phagosome. In contrast, the other process that we're more interested in studying is mediated by a specialized secretion apparatus called a type 3 secretion apparatus, and I believe Arturo has mentioned it before, that leads to actin polymerization, not only in close contact, but at a distance from the bacterial surface, as we'll see. And this would lead to the engulfment of the bacteria in a wide vacuole. So type 3 injectosome, you probably saw this picture several times already, just to mention that not only they act as they function as injector of macromolecules inside host cells, but they also look like needles here based on 3D reconstruction of single particles. And you can also notice that there's only discrete members on the bacterial surface as viewed by electron microscopy here. And it's been estimated that between 50 and 100 copies are present on the bacterial surface. So I think Arturo has mentioned the tip complex, and I think I will mention here also the various components of the type 3 apparatus. So you have here the basal body that consists of 2 stacks of plates, and the assembly of this basal body is dependent on the sec apparatus. And then we have the needle here that is made of the MXIH protein, and the tip complex, which consists of IPA-D mostly, and perhaps IPA-B as well. So upon cell contact, IPA-D and IPA-B and IPA-C are secreted, actively secreted, and insert into host cell membrane, where they form a so-called translocator, or a pore, through which other injected effectors are translocated. And this is these translocated effectors that would lead to the cytoskeletal arrangement that lead to invasion. So the model we're studying is Shigella, the agent of bacillary dysentery. And as you know, Shigella is able to invade epithelial cells, lyses the phagocytic vacuole. Once inside the cells, it can replicate. And during this replication phase, it polymerizes actin at one pole of the bacteria and can move intracellularly, pushing protrusions into adjacent cells and moving, therefore, from cell to cell without extracellular step. And it is believed that both invasion and dissemination are important for bacterial virulence. So the nice thing about working with Shigella is that it's very close to E. coli, so it's very amenable for molecular manipulations. Also, genetics is available. And the genome has been sequenced. And the big difference between Shigella and E. coli is the presence of a large virulence plasmid. And thanks to the input of genomics, but also of several studies, we have identified most of the type 3 effectors that are injected upon cell contact. And those can be subdivided into classes, those that are constitutively expressed, and injected rapidly upon cell contact. And those, you can imagine, would imply those are involved in invasion, such as the EPA proteins, and other effectors that we described, for example, in the inhibition of autophagy or the inhibition of macrotubule polymerization. The second wave of effector, which we won't talk about, are expressed, upregulated after injection, so we believe that they're expressed only once the bacteria is inside the cells, and those involved in the modulation of the immune response. So this movie here just is a phase contrast, dual acquisition with SARC-GFP. And you can see that upon cell contact, the bacteria initiates the recruitment of the SARC tyrosine kinase, and which leads to the engulfment not only of one bacterium, but at several bacterium at the initial site of interaction. So we can see by filming here that actually invasion of one single bacteria would be proficient, would be used by several other bacteria to get inside cells. And we believe that this is important for further processes. So you can see also that during the early stages of contact, you have recruitment of the tyrosine kinase at the intimate contact, which leads to speculation, or which makes us believe that during the initial contact, recruitment of the Src kinase is important for invasion. So to cut a long story short, I would just resume what we think is happening during invasion in terms of interaction between type 3 effectors and signaling responses. So as I mentioned, the B and IPA-B and IPA-C are inserted into the whole cell membrane, and IPA-C not only acts as a translocator, but also signals rapidly. And it signals by activating the CDC42 and RAG GTPases that lead to actin polymerization through ARP23. And also, and as we'll see, through the recruitment of Src that would amplify this actin polymerization from the intimate contact to the distance, leading to formation of ruffles that engulf the bacterium. Concerning the effectors, so here are in red are the infectors that have been identified as playing a role in bacterial invasion. I mentioned IPA-C. We also know that IPA-A, for example, is important for actin depolymerization, and this is important not for the initial phases of bacterial entry, but for the completion of the entry process. And similar things occur during phagocytosis. We need to downregulate actin polymerization to have efficient phagocytosis. The other effectors is IPGB1 that's been involved also in the recruitment of a GEF, an exchange factor for RAC, which is the Elmo dog, and thereby amplifying as well actin polymerization from the initial site of actin polymerization. Other effectors such as VRA, for example, are involved in microtubule inhibition, and it is believed now to play a role in actin-based motility, while others like IPGD, for example, is playing a role in the hydrolysis of PI45P2 to PI5P, and that could be involved either in facilitating bacterial entry or reprogramming the cells to modulate inflammation, or cell survival, excuse me. First, have cues about how IPA-C signals actin polymerization. We treat cells here with an inhibitor that inhibits actin polymerization. So why do we do that? Because as you saw from the movie, entry foci are very dynamic, so it's very difficult to have a better idea of component recruitment unless you freeze the system. And one way to do this is to inhibit actin polymerization. And when you do that, you can see that you can still see patches of actin, and you can still see here the recruitment of Src where the bacteria is, indicating that you can recruit the Src kinase in the absence of actin polymerization, suggesting again that event of Src recruitment is a very early event during bacterial entry. So not only we can see recruitment, we can also see that IPA-C, which is this translocated component that we stain here by fluorescence, colocalize with these patches of Src that is coating the bacterium, suggesting that perhaps IPA-C plays a role in Src activation. So one difficulty here, as you can imagine, is to be able to distinguish between a role for IPA-C as a translocator or a role for IPA-C as a molecule that signals to the cell. So one way to do that here is to identify mutants of IPA-C that are not affected for the translocation of type 3 effectors, but that are affected for signaling. So here, for example, this is the domain organization of IPA-C with the transmembrane region, the chevron binding site, the secretion signal, and in particular in the C-terminus, the last 63 cubic C-terminal residues, a domain that has been identified by we and others, like the effector region responsible for actin polymerization. So in the lab, Joël Mounier mutated this region to identify key residues that are involved in actin polymerization, and she was able to identify mutants that were not able to induce actin polymerization, and yet were still proficient at inducing injection. So using this mutant, this suggested that IPA-C was involved in Src recruitment and actin polymerization. But is this sufficient? So one way to do this is to fuse the same domain to a delivery system. Here we use the iota toxin, which is close to the anthrax toxin, that is based on a B component and an A component. So the B helps binding of the A and translocation of the A component, the catalytic subunit, that we just use here to use to translocate here the fusion protein inside cells. So if you fuse here the C-terminus domain of EPA-C to this A component, you can see that you can recapitulate actin polymerization foci in cells that are simply treated with recombinant IAC. And those foci are virtually indistinguishable from those that are induced by Shigella upon invasion. Those are very rare structures. So we believe that here EPA-C is clearly a key factor in inducing actin polymerization. So not only it induces foci formation, but also induces the recruitment of the Src kinase, of those ruffles, and of those foci that are induced with this recombinant protein. Either Src or activated Src as observed using an antiphosphatyrosine antibody that recognizes only the active form of Src. So interestingly, if this time you don't use you don't stain for EPA-C, but you stain for another injected effector, such as EPA-A, for example. There's been triple flag to enhance the visualization. You can see that EPA-A is clearly translocated upon cell contact here. Staining is in red. But if you inhibit actin polymerization, you limit as well the injection of EPA-A at one pole of the bacteria. So not only injection of the effector is altered in terms of quantity, but also you can see that the initial site of injection seems to occur through the pole. So that would tell us that during entry, basically you need EPA-C to activate actin polymerization. And it seems that actin polymerization is also important for good translocation of effectors. So in other words, we believe that actin polymerization could help, for example, establishing contact between the plasma membrane and the type 3 tip complex, thereby allowing further translocation and more efficient injection of effectors. So in other words, for a long time we're distinguishing between the trigger and the Ziegler hypothesis, the model. So it's possible that the type 3 also involves some type of hybrid, in the sense that you need to signal to inject to signal, excuse me, also to signal to inject. And you will have some type of Ziegler model whereby you need to have an incremental effect of the type 3 effectors to get efficient. So changing perspective now, we'd be interested also to try to understand how a bacterium that inject effectors can induce actin polymerization and other effectors that do exactly the opposite, EPA-A, and the combined effect of those two effectors are important for bacterial invasion. How are they coordinated? One way to do this is to visualize the injection effectors, and this is what Joost and Inge in the lab had done a couple of years ago now, where he labeled the effectors with the flash protein. And he did that because we knew that fusion to GFP, which is the green fluorescent protein that is widely used for imaging purposes, was not good for studying type 3 injection because it would clog the type 3 secretory apparatus. And this has the advantage that the epitope here is very small. You can incubate the cells with the arsenate fluorescent derivative now and then label this effector fluorescently. And by doing so, you're able to see upon staining the injection effectors inside those entry foci. For example, if you focus on the bottom panel here, you can see that the bacteria here has injected EPA-B, which is flashed in entry foci in green, while in other foci that seems to be perhaps more early events, you have a lesser injection of EPA-B. The big advantage of this approach, however, is that you can use real-time imaging live cells to image type 3 secretion, not so much in the appearance of the fluorescent effector inside host cells, but more the disappearance, just for technical reasons. And you can see, for example, that this bacterium here sees its fluorescence decreasing over time, actually very rapidly. And if you average several of those events here, you can see that secretion occurs upon cell contact. So this is what has been speculated for a long time, but now we can eventually see it. Secretion occurs upon cell contact and very rapidly. So you can see that after a couple of minutes or 4 or 6 minutes, you have almost virtually complete secretion of the type 3 effectors. So in other words, it seems that type 3 secretion is highly efficient and is induced really upon cell contact. So this is one aspect also of type 3 secretion that one has to take into account when thinking of its role in bacterial invasion. The other aspect, and this I referred to a couple of slides ago when I showed EPA-A secretion in the presence of inhibitors of actin polymerization, If one looks at HEPA-C this time, which is this translocator component, at the very early stages of bacterial invasion, you can clearly see that HEPA-C is also secreted at one pole of the bacterium, here in red. And Valentin Joumouillet, who was in the lab during his thesis, has shown that this polar secretion of HEPA-C was correlated with the observation that HEPA-C inside the cell also localizes at one pole of the bacterium. Here will be GFP, for example, and this is HEPA-C GFP that labels only one pole of the bacterium. So not only it labels one pole of the bacterium, it is precisely this one pole that is proficient for type 3 secretion. So here you have HEPA-C GFP, and here you have staining of HEPA-C after secretion. And you can clearly see that this is this one pole that is being labeled that is proficient for secretion. So how did Valentin do that? Because I just mentioned that HEPA-C GFP was blocking secretion. Well, just by playing with the amount of HEPA-C GFP, reducing the amount using an inducible promoter to such an extent that you could still see type 3 secretion. So more than this, if you use several fusion of HEPA-C this time that affect the localization of HEPA-C within cells, we know, for example, that some truncation of HEPA-C would result in diffuse localization, while some would lead to intermediate localization. It appears that the localization of HEPA-C seems to correlate also with the efficiency of type 3 secretion. In other words, if HEPA-C is polar, such as wild type, secretion is very efficient within 5 to 6 minutes, as I mentioned earlier. And if you have a truncated derivative that is also polar, you have the same kinetics. Whereas if you have a diffuse HEPA-C, then this substrate is not secreted anymore. So this is highly exciting because we believe that this polar compartment here at one pole of the bacterium seems to regulate the efficiency of secretion, and that the bacteria, the substrate, need to polarize at one pole for efficient secretion to occur. How does that work? I think there's a whole field of bacterial microbiological physiology that needs to be investigated in terms of its relationship with secretion. So we'll also be interested in how this is one of the reasons why I got interested in the formation of this pole here. And this is just time-lapse showing the accumulation of HEPA-C GFP, or variant of GFP that falls rapidly over time. And you can clearly see, if you focus on those bacteria here, that the dot appear at the onset of bacterial division. So the bacterial septates, and you have one dot that appears directly at one old pole here, or a new pole on the other side. So that clearly, see, old pole, new pole here, and the old pole. So that clearly suggests that the formation of this polar compartment is linked to bacterial division, and perhaps the processes that are involved in the polarization of the substrate here are the same, or at least some of them are the same, that those are involved in bacterial division. So eventually I just want to show you the slide here. Again, this is Shigella that induces actin polymerization. You can clearly see that actin polymerization at the very early stages occurs at one pole of the bacterium. We hope that in a few years from now we'll be able to see things, to image the interaction between the effectors and the components, how they organize, how those in real time recruit the SAR kinase, lead to actin polymerization. So it's a bit like science fiction right now, but you know, if you think about it, perhaps it will come. And just to resume, we've shown that type 3 secretion occurs within minutes upon cell contact, and that the whole pool of the type 3 effectors can be secreted within seconds. That the type 3 secretion occurs predominantly at one bacterial pole, and this secretion is determined by intrabacterial substrate polarization prior to secretion. So in other words, that brings another dimension that we would not have if we didn't image the invasion process. For a long time, we're studying interaction between effectors and targets. One possibility here is in the order for type 3 secretion to be so efficient, invasion to be so efficient, you need to focalize secretion at one point of the bacterium, at the point of contact, therefore, and have a very efficient type 3 secretion system so that locally you can reach very high levels of effectors. In other words, you would focus the concentration of effectors with a high intensity. And these two factors, these parameters, might be very important for the triggering of the entry process and of actin polymerization. So I'll just finish here by thanking people who did the work. Sorry, Jost, I think you will have to send me a new picture because I keep selling this one for a long time now. This is Jost when he was in the lab. I believe he will give another presentation later in the afternoon, so you have a chance to see how he has changed ever since he was in the lab here. So he worked on the flash system. Joëlle Mounier, I regret that, anyway, she was in Philippe's lab. We have been collaborating for a long time, and I was very happy to collaborate with Joëlle, and a bit sad that it's over now. And of course, this work was done in Philippe's lab. And Valentin was also a PhD student in Philippe's lab. He's now with me at the Collège de France because I moved here about a month ago, where we'll try to pursue some of the work while expanding perhaps some of the thematics. We have more lab space now, so if any of you have people you can send me, good people, I'll be willing to receive them. And so this work has been financed by, of course, the Institut Pasteur, the INSERM, Collège de France, the DNR, and in part by the NIH. Thank you.
Thank you very much, Guy. We have time for a few questions.
Yes, so perhaps this is exactly the point that we're trying to make in Valentin's paper, in a sense that there are several ways to explain type 3 secretion. So one of the way we believe in the type 3 is polar localization. It's exactly the same process that's been suggested for ICSA. ICSA is supposedly sec-dependent to cross the membrane, but inside the bacteria is polar. So it's intra-bacterial polarization that determines polar secretion. So in the case of the type 3, you can imagine that there might be type 3 secretion all over the bacterial surface, but because Epithelial is mostly polar, secretion will take place at this one pole here and occurs predominantly at this one pole. And I just pulled down the same type 3 secretion real-time imaging that Jost performed, because we observed at the time that once in a while you can see that in some bacteria you had secretion starting and then stalling and resuming again. So that's interesting because that suggests that there might be discrete events in type 3 secretion. So one way to interpret that, for example, would be that perhaps there's only one single apparatus that is active at a time, and when it switches off, then secretion stalls. This is one possibility, or perhaps a discrete number of apparatus. I think we would be very interested to determine what. Sorry, we're still talking what I was answering. I think this will be very interesting to challenge. How many active type 3 effectors is this sufficient to convey the secretion of the whatever molecules in 4 or 5 minutes? I think this will be highly interesting. We know it's a very efficient system. How many molecules can you inject through this effect to the system? I think this will be proper in vitro modeling.
I think one issue which we have never addressed is, when we speak about lack of polarity of type 3 secretion apparatus, this is in vitro situations. We've never seen in vivo if there would be some clustering or recruitment of more secretory apparatus at the site of initial interaction.
Recruitment like this, you mean?
Right.
I think, yeah, it's possible, but I think, because those apparatus span the wall, I think it's very unlikely that they diffuse rapidly.
You know if IPAC interacted with any of the cytoskeleton protein inside the bacterium, Vidaxin or FTS?
No, we don't know, and this is actually one of the projects we'd like to pursue with Clémence in the lab. So we'll try to identify which component it is. It seems that FTZ in an FTZ mutant is still polar, and in the MRB mutant, sometimes you make patches as well. So it seems that it's not critical for polarization.